Hello, everyone. Thank you for tuning in to Manav Data Science webinar series. Today, we are going to do something very interesting. We are going to look into a phenomenon which is billions and billions of years old through a new perspective which mankind acquired just a few decades ago, or maybe a century ago. Yes, you're right. We are going to look into evolution through the lens of computational modeling. Hello, I'm Dr. Preeti Nema. I welcome you to 36th episode of the Manav Data Science webinar series. Today, we are privileged to have with us Dr. Supreet Saini, who is the professor in computational biology department at IIT Bombay. Dr. Saini is an intermediate fellow at the DBT Wellcome Trust India Alliance and the professor at chemical engineering at IIT Bombay. He completed his BTech from IIT Delhi and his PhD from University of Illinois, UC. His research group works on understanding speciation events using the yeast model as the model system for evolution. Title of his talk today is Natural Selection in Action in Laboratory Experiments and Ecology. He'll be talking about how natural selection acting on the genetic diversity in a population selects for a beneficial mutations in a given environmental context. In his talk, using examples from laboratory experiments and ecology, he will discuss how an evolutionary process shapes the evolution of a species. Before we begin the talks, a small announcement. A certificate of participation is available for this webinar. Please fill in the feedback form, which will be made available to you at the end of the webinar. And please put in your questions in the Q&A tab. If you are attending this webinar through the Manav YouTube channel live streaming, please put in your questions in the comment section and we'll take it from there. So without any further ado, I'll hand over the mic to our speaker for today. Over to you, Dr. Saini.
Thank you, Preeti. It's a pleasure to be here. I will share my screens, and if somebody could just confirm if you can see my slides.
Yes, it's visible, sir.
All right, thank you. Great. So what we'll do for today, I thought, is a whirlwind tour of evolution, and obviously it's a subject that has fascinated people for thousands of years. But really, our intellectual breakthrough has been made in the last 200 years or so. And more than anything, as a student of evolution, what I really enjoy doing is studying the history of the subject. And how we've come to understand the process called evolution the way we do today. What I'll do today is sort of, I'll also try and communicate the excitement I have for the subject. And I think the best way to sort of communicate that excitement is showing you some of the experiments, some of the really neat experiments that people have done. So this isn't really going to be about the work that we do in our lab, but some of the studies that I think are really cool and have enhanced our understanding of evolutionary processes as we understand them today. Should you have any questions about anything I talk about or anything that you would like to run by, everybody is very welcome to write to me at the email address that I have here. All right, so let's get started. Life is about, our planet is about 4.5 billion years old, 4.6 billions. And what this arrow shows is the time from the leftmost edge is the time when the planet was formed. The rightmost, the head of the arrow is where we are today. And life has gone through some dramatic events in order to bring us to the place where we are today. And the first evidence that we have for life on Earth is actually very, very old. It's close to 3.6, 3.7 billion years ago that we have first evidence of life on the planet. For the next about 1.5, 2 billion years, these single-celled prokaryotes without nucleus were the only life forms existing on the planet, bacteria and archaea. And it's only about 2 billion years ago or so that we have the first eukaryotic cells come into existence. And once that happened, it took a further billion years or so for the first multicellular organisms to come up. And the first evidence we have that is from an era called Ediacaran fauna, which happened about 560 million years ago or so. This was followed by, unfortunately, the organisms that were there in Ediacaran fauna did not last, and they all went extinct fairly soon afterwards. But that led the ground to what is called a Cambrian explosion, in which there was a diversity generated as there was never before on Earth in terms of the life forms and the organ systems that evolved in this period. Cambrian explosion is one of the most fascinating areas, most fascinating periods to study of history of evolution on Earth. Then about 200 million years ago is the largest extinction event that this planet has seen, which is called Permian extinction, which led to more than 90% of the species on the planet being wiped out. This incidentally also created the space such that the ancestors of dinosaurs were the prevailing organisms, and this led to evolution of dinosaurs. About 65 million years ago is the other big extinction event, which is called the KT extinction period, which led to the dinosaurs becoming extinct, again creating space for newer species to come into being. And really, our existence is just a tiny speck on this time spectrum. And they say that one of the things that we have trouble imagining is the immensity of the time that this arrow poses. And I think 4 billion years ago is something that we say as a number, but it's very difficult to get a hang of what that means. So as I said, that our understanding of evolution as a process really took off in the 20th century. And while there were many people who worked and enhanced our understanding in the first half of the 19th century, really modern evolutionary theory is founded on Darwin's work. And Darwin was born in 1809, and when he was 22, he went on this ship voyage on a ship called HMS Beagle, which took him around the world and was there in the ship for 5 years. Most of the time was spent on the east coast of South America, just mapping coordinates. And the famous Galapagos Island, where he saw finches, are off the coast of South America here. This is off the coast of Ecuador. And this is, along the way, he's a meticulous note keeper. He's making notes, he's collecting samples, he's constantly sending samples back to England. And by the time he gets back to England in 1836, he's one of the most celebrated naturalists in all of Europe. And surprisingly, even though The Origin of Species, the book was published in 1859, so 23 years after the voyage had ended, as soon as the year following his return to England, Darwin, in one of his private notes, his notebooks, this is a very iconic image in study of evolution, where in 1837, in one of his notebooks, he makes this sketch and draws this tree of life. And he, from all his observations, note keeping, and conclusions about how life could have come about, he thinks that the only natural way to represent how life forms have come into being is this tree-like arrangement, where we have a node, which is one species, which splits into two different nodes at the end of these arms, and which is two species, and hence diversity is therefore generated. And he says that the only natural arrangement of life forms that could make any logical sense is this tree-like arrangement. Of course, the popular opinion at the time was that life forms are immutable and have been created in their present way, shape, and form the way we see them, and have existed like that forever. But Darwin could see that that wasn't true. Species were in fact undergoing change all the time, and hence this tree-like arrangement. This tree-like arrangement has actually gone several changes from Ernst Haeckel's tree to Robert Whittaker. Carl Voss made a gigantic leap in the 1970s and 1980s. But really, our modern understanding of a tree can be captured by this cartoon that was published in this review for 20 years back. And the life originated when so the common ancestor to all existing life forms is called last universal common ancestor, which is also called LUCA in the field. And this LUCA then evolved into two different life forms. One was bacteria, which are called true bacteria, also called eubacteria. The other one was archaea, which were at the time called archaebacteria. And these two life forms existed for a couple of billion years. And when one day an archaea engulfed a bacteria, and that bacteria subsequently became the mitochondria, and hence this led to the rise of eukaryotes. And then all of these eukaryotes have come into being much later. And this engulfment event, where archaea engulfed eubacteria, led to the evolution of the first eukaryote from which all eukaryotic life has evolved. And this first eukaryote is called last eukaryotic common ancestor, and is the ancestor of all eukaryotic lives that we see around us. And again, from a computational perspective, we don't have LUCA and LECA to study in lab. So all we can do is understand, do a lot of genomic analysis and try and understand what were the physiological and metabolic capabilities and what were the genetics of LUCA and LECA. And a lot of work goes on into understanding these extremely these events that took place in the deep past, and try and understand that how, starting from such humble beginnings, life in its present shape could have evolved. All right, so Darwin proposes theory of natural selection, and at least in a scientific context, there isn't much of an uproar after the publication of the book. So if you try and understand the theory of natural selection, we need three elements for natural selection to act. The first one is that in a population which comprises of a number of individuals, variation must exist. All individuals cannot be identical. If they are identical, then we cannot have natural selection acting on it. And these different colors here represent individuals with different capacities. We can think of this as bacteria with different morphologies, some difference in one of the aspects of their physiology. Because they are different in the prevailing conditions, each one of them will reproduce an unequal number of times in a given interval delta t. So if we look at this, if we look at these 5 individuals after time delta t, this one has gone to 1, this one has still not reproduced, this one has divided once, this one has gone up to 3, and so on and so forth. So what this means is that this variation was then acted upon by natural selection because their fitnesses or the rate at which they were dividing were different. And hence, although the blue individual here formed 20% of the population, by the time delta t time passed, the blue individual is now 1/8 of the, or 1/7 of the, or 1/8 of the population now. So from 20%, it's gone to 12.5%. And hence a change in frequency of the blue individuals has taken place. And evolution is essentially this, that if different types of individuals increase or decrease in frequency, that's evolutionary change. And the third component is that this variation that was present in the population is also inherited. So the blue individual gives rise to a progeny, which also is blue in nature, and so on and so forth. So what we have seen, these are the 3 key elements of the theory of natural selection. And what we see here is that natural selection needs variation to act. If they were all blue, then we had 100% of the individuals blue, and all of them would reproduce to produce blue progeny, which means 100% of the next generation will also be blue. So there is no change taking place. So natural selection needs variation to act. But what we must realize is that while natural selection needs variation to act, natural selection also reduces variation. There were 5 variants here, and because this individual was slow, it was wiped out by competition, and now there are only 4 types of individuals. So variation in the population has actually gone down as we progress from the ancestral generation to the current generation. And hence natural selection is a force that needs variation to act, but it also consumes variation in the process of its action. So as we see, if we keep on going forward, eventually this blue individual is also going to be outcompeted, because we see that these 3 types of individuals reproduce much faster. And subsequently, this gray one will be the only types of individuals left in the population. The others will be beaten by the competition it offers. So then how does new variation come into being? And obviously the fodder for new mutations is mutations happening, which is the error in the DNA replication process that happens during the process of copying of DNA in preparation for cell division. So what I'll do now is, so that these are the core elements of the theory of natural selection. And what I'll do today, or what I'll do for the rest of the talk, is give you a sense of how we can design experiments to understand this evolutionary experiment, and how we can infer from what is happening in ecology and use data available from ecological samples and mathematical techniques to infer consequences about the evolutionary processes acting. So let me show you 3 types of evolution experiments which were done in lab. So the first one is this paper that came out in Science a few years back, and I'd like to show you guys a video. Again, if somebody could just confirm if the video is up. Can you see the video?
Yes, we can.
Excellent. All right, thank you. So this is a study that was done at Harvard in Roy Kishne's group. And what this is really doing is, what they've done is this made this giant petri dish. We've all seen petri dish which we can hold in our hands. This is just a giant version of that, which is 2 feet wide and 4 feet long. And what they do in this study is that they divide this giant petri dish into these chambers, and in the leftmost window they have no antibiotic. In the chamber next to it, they have a little bit of antibiotic, then 10-fold amounts of that, 100 folds, and the middle chamber has a 1,000-fold of the lowest amount of antibiotic that was present. This lowest amount of antibiotic is enough to prevent growth, such that the bacterium that they are starting with will not be able to grow in this amount of antibiotic. And the right side of the plate is just a mirror image of what we see, what we have on the left side. So the plate is symmetric left to right. All right. Then at the start of the experiment, what they're doing is, now they will allow the bacteria to grow from left and right. So I won't say right, because right is just a symmetric mirror image. They will allow growth to take place from left. And what's important is to realize that on the leftmost side, there is no antibiotic for some distance, and hence they're growing fast. But see, now they've hit a roadblock because they can't move forward because there is antibiotic and they can't grow. So what has to happen now is that a mutation has to take place at this interface somewhere. Some individual has to pick up a mutation such that it can survive and propagate in this antibiotic concentration of 1. And you don't actually have to wait very long, and you see these mutants rise up. The one on the right is very prominent, but you see several coming up on the left as well. And as soon as they rise, it spreads very fast and takes up the and occupies the space that was available in the window, which was 1 unit of antibiotic. But if you see, it again halts, and it needs to pick up these new mutations, which are these fresh strains that you see coming out, to be able to survive and grow in the section of the plate which is antibiotic 10. And this keeps on repeating itself, and it occupies concentration 10, and then you again see new mutants coming up on this plate, and all of this is happening over a time scale which is of the order of I believe it's 1 to 2 weeks, so not very long. So if you see, this is an excellent demonstration of how antibiotic resistance can evolve within a matter of days in a sample size which is actually not very large. This is just a petri dish of bacteria, but resistance comes up very, very rapidly. And what's interesting is that when we noted the first interface that it hit, it wasn't as if there was one mutant which was able to grow. Several mutants arose along the interface, and they all started growing in the region containing the antibiotic. And statistically speaking, it's very unlikely that it was the same mutation. And hence, what we understand from an evolutionary process is that now several mutations in the genome, it is possible for me as a bacterium to acquire mutations in several parts of the genome and become resistant to the antibiotic. So studying this and the statistics of this evolutionary process is what this science paper is all about. All right, we will go back to the presentation. So this is an example. So if you see these nodes, this is a picture from the same video. We can now trace back and understand that what mutation happened where and how did it spread going forward and so on and so forth. So this offers us the statistics of the evolutionary process, which is actually quite a remarkable area, because evolution is a statistical process. Every time you do an evolutionary experiment, the outcomes are going to be different from other tries. And hence this study offers an insight into that aspect of evolution. The second study is the longest running evolution experiment that I'm aware of. It started in the year 1896. It still continues. This is an oil field. This experiment is not being done in a lab setting, but it is being done in an agricultural land. It's a corn oil experiment in Illinois in the US. This is also the place where I did my graduate work. What they do in this study is that every year they have a field full of corn plants, and they take corn from each of the plant and measure how much is the oil content in each of the plants. And then there is a threshold, and so there is a threshold of, let's say, 10 to 15%. Only the plants containing the highest amount of only the top 15 oil containing plants are allowed to replicate and form the next generation. The remaining 85% are just discarded every year. So what goes in the next generation comes from the top 15 oil content plants for the previous generation. So only these guys are allowed to replicate and form the next year's generation. And in the next year's generation, the same process would be repeated, that only the top 15% are allowed to form seed for the following year's generation, and so on and so forth. And this process has been going on for over 125 years now. And what we can see, forget about rest of the lines. I only want you to focus on the dark green line. In 1896, the oil percentage was about 5%. Today, this line has gone up to somewhere between 20 and 25%. What is remarkable here is that this increase has happened not in a stepwise function, but it's the linear fit is a very nice fit for this process. Conventional understanding of an evolutionary process would be that oil content would only increase when there is a new mutation that comes up, and that mutation leads to increase in oil production, and hence it spreads in the population. But that's not what we see. If that was the case, we would wait and see no increase in oil production, and then there would be a step increase, and then it will flatten out, and wait for the next beneficial mutation which leads to increase in oil production to come up. But that's not how it proceeds. Evolution proceeds in terms of increase in oil percentage in a continuous fashion, in a linear fashion. This again leads to excellent insights into the genetics of the system, and what is the genetic control of oil production in at least how corn behaves in terms of a plant. And again, both theory and experiments help us understand what's going on in this system. The third lab experiment that I want to talk about is actually the longest evolutionary experiment in terms of number of generations that are processed. The previous one that I talked about is the longest that I know of in terms of the time that it's been going on, 125 years now. But in an evolutionary context, the relevant time scale that we are interested in is not how long the experiment has been going on in terms of magnitude of time, but in terms of how many generations have we processed. So in this corn experiment, we have only processed 125 generations, whereas in this evolutionary experiment with Ecology, which has been going on since 1988 February, we've processed more than 75,000 generations. And in these 75,000, and hence the wealth of data that has been generated through this experiment is just humongous. And this evolution experiment, so I've already shown you two ways to do evolution experiment. This in this third way, and this is also the most common way of doing evolutionary experiments. What you do is you grow cells in a flask in any given environment that you are studying. In this particular environment, they are studying how E. coli evolves when glucose is in very short supply. So glucose is the carbon source in this experiment, but there is not a whole lot of it. So the cells have to adapt to become better at utilizing glucose. And what happens is that they will soon exhaust all the glucose that is available here. So what you do is you take 1/100th of all the cells present here and transfer them to the fresh flask, which is containing the exact composition as the starting flask did at t equal to 0. And you keep on doing this every single day. This experiment has been going on for, well, now more than 30 years. And we doing an experiment like this, you process, again, we can do some math and understand that you process 6 to 7 generations per day. 75,000 generations is a lot of generations. And there are over 200 publications from this experiment alone in these 30 years that have passed. But what this has really told us about is that what is the roles of chance and determinism in evolution. I just went through your lecture series, and I saw that sometime back Kavita from JNC talked about the chance of the role that chance plays in deciding evolutionary outcomes. So this experiment has been an excellent model system to understand that, because when they did the experiment, they did not just do this experiment once, but there are 12 independent and identical lines. This experiment is being done a dozen times independently in parallel. So flask 1 is being propagated every day, flask 2, and so on and so forth till flask 12. So do all 12 lines behave identically or they diverge is something that is of interest. And we have learned a lot about this particular question from this experiment. It also leads us to a question of repeatability in evolution. One of the books that I would recommend everybody, all of you interested, read is Stephen Gould's. He was a paleontologist. Stephen Gould's Wonderful Life, and in that he asks a rhetorical question that should we go back 4.5 billion years back and start evolution again on this planet? Would it be organisms like you and me who would be inhabiting the planet 4.5 billion years again if we were to just redo Earth from t equal to 0 again? And Gould calls it replaying the tape of life, that if we replayed the tape of life, would it follow the same trajectory or would it just completely go off on another trajectory? And while that's obviously a rhetorical question and not possible to do in any level of complexity, this experiment gives us insight in the sense that it's evolving E. coli in an environment, the organism that we understand the most, in an environment, and it's doing it 12 independent times. So we can track them with time, and we can see that are all 12 independent repeats of the same experiment going towards the same evolutionary outcome, or are they going towards evolutionary outcomes which are just very different from each other? And around the generation, I believe it was around generation 30,000 or something, one of the 12 lines actually started doing something really strange, which is considered to be an example of metabolic innovations. How do metabolic innovations arise in an evolving population? And that particular innovation can also be looked at in the context of a speciation event happening in bacteria, where you can pose the question that is this species that is evolving in this particular flask, is that even E. coli or has it become something else? So again, a wealth of data from this experiment, but these three are sort of among the many evolutionary studies, ones that stand out. What we want to do is is view this process in a geometric context. There was this framework that was proposed by an American geneticist, Sewall Wright, in 1932, which is called fitness landscape. And the way you can think of it is, imagine that you have a gene sequence which encodes for a protein, and this protein is really important, and you have 4 such variants. one of the codons is ATG, and ATG's fitness is 1. An individual which is carrying this particular sequence, the orange one, has a fitness 1. An individual carrying ATT, which encodes for isoleucine, has fitness 1.15. AAT is red, is 1.2, and AAA is lysine, 1.22. So if you see, as I transition from orange to blue to red to green, fitness is increasing. And what can happen is that if my starting population is all ATG, then all my individuals are growing at fitness 1. But what would happen is that one of the individuals might acquire a mutation such that this G becomes a T. If this G becomes a T, then this individual is suddenly now going to grow at a fitness of 1.15. So while if the population size is 10 million, 10 million minus 1 individuals are still growing at fitness equal to 1, but this one individual is growing faster now, and hence competition will competition will mean that this individual now is able to eliminate the others from the population, and now the population moves to a fitness level of 1.15. But the same fate will repeat itself till when one of the individuals carrying ATT picks up another mutation, and this ATT becomes AAT, and now you have rest of the population at the blue node, but one of the individuals has moved to the red node, and hence this red individual will now outcompete the population at blue, and the population now moves to red, and an evolutionary process continues, so on and so forth. So in this process, in this view, evolutionary process is akin to climbing a mountaintop. That at any node, you are looking for any mutation which leads you further up on the fitness scale, and hence evolution is often viewed geometrically as climbing a mountain. However, this mountainous view becomes very difficult to look at because of the dimensionality of the problem. Because right now we are only looking at changes at this one particular location. How do we mix this when we are also talking of changes at this, and genome sizes are very, very large? A typical bacteria's genome size is of the order of 10 to the power 6 bases, and here we are looking at only 3. So this also has interesting implications, and this is a subject that has been studied computationally in great detail. That what is the relationship between the genotype of the individual and the phenotype of the individual? If the genotype-phenotype relationship is of a certain kind, and we won't get into the details of what kind, then the mountain that we are climbing, then the mountain that the population is climbing is a smooth one. And this geometric view is called a fitness landscape. This is Frances Arnold's images from Frances Arnold's paper, who got the Nobel Prize a few years back. This is a review that she wrote in 2009. So if the genotype-phenotype relationship is of a certain kind, then we have a very smooth mountain. Now this has evolutionary implications, because what this means is that if your starting population was at the left end of this graph, it will pick up mutations and climb the top and will reach the top of the mountain. But if your starting population was on the extreme right of this graph, even then it's climbing the same mountain hill and it will reach the same peak. And hence the evolutionary outcome of the process will remain the same no matter where you start on this three-dimensional graph. On the other hand, if the genotype-phenotype relationship is slightly complex, and that complexity is introduced by a phenomena called epistasis, which we will not talk about today, if the relationship has epistasis, then you see these multiple peaks arise. And then, as shown in the two lines here, the red line and the green line, even though you are starting from the same position, the populations can end up on two different peaks. And hence an evolutionary outcome, the evolutionary outcome of a population is also dependent on chance, that which direction did you end up taking. And if this phenomena called epistasis is very high, is extremely prominent, then you have a fitness landscape which looks like the picture that's shown on the right, which is just deep valleys and extremely sharp peaks, and a whole bunch of them. And obviously evolutionary outcome in such a context becomes a highly noisy process, a very chance dependent process. And this is one of the factors which brings in chance that I believe Kavita must have talked about whenever she was here. And all of this, while this is being realized in 2009 experimentally, in the last decade or so, we've been doing experiments and theory to confirm some of these things. This was actually predicted by Sewall Wright in 1932. And this is a paper from this is an image from his paper, where the pluses indicate peaks and the minuses indicate valleys. So Sewall Wright viewed evolution as movement on this terrain which is full of peaks and valleys, and that is very, very much something that we're still scratching the surface of, even after close to 100 years. So this slide is dense. I don't want to get into the detail, but I just wanted to communicate that theory and experiments are done to understand what is the real nature of landscape. I mean, the top three, Francis Arnold's pictures, they're just cartoon representations. We don't really know for sure what do these structures look like. Theory has given us some insight into it, but really we don't really understand what do these landscapes look like. And in the last 10-15 years or so, there have been these two very nice studies. One is from Lenski's group, and the other one is from Daniel Hartel's group. And they study two different systems. But the point that I want to communicate here is that when they study this system in 2011, they found that the landscape, the geometry of evolution looked like as if you were moving up a very smooth hill. So the landscape is largely smooth, and hence you are all climbing towards the same hill. However, in the 2006 paper, which came out several years before this, what they showed was that the movement is actually not smooth at all, and landscapes look like these jagged peaks and valleys. So we don't really know in terms of really being able to understand either via theory or experiments that what do these landscapes look like. We're only just beginning. I mean, since these papers came out, there have been some more examples in support of both these types of structures, but there is a lot to be done in that direction. All right. Last few minutes, I will talk about the fact that while these three examples and these last two studies that I talked about, they all talk about evolution in an experiment which is very carefully controlled. So your flask every day in Lenski's experiment for 75,000 generation, the flask every day has the exact same amount of salts, the exact same amount of glucose, the temperature is never varied, the rotation speed of the flask is never varied. It's a very, very carefully controlled environment. And we do that for a reason. We do that for a reason because we don't understand evolution well enough to be able to generalize, to be able to understand it in an environment which mimics what is perhaps out there in ecology. So compare and contrast the environment in which Lenski's E. coli is evolving for 75,000 generations with the evolution that is taking place in a soil in a garden near you. That soil sample goes extremely dry. So all the microorganisms there have to survive that dry patch. Then it might get flooded. Then temperature increases during the day, decreases during the night. During the winters, it's extremely cold. Well, if you're up in the north anyway. During summers, its temperature is high. There are lots of predators around. There are lots of fungi which are producing antibiotics. None of these fluctuating parameters are present in any of these lab studies that we want. And my reason for bringing that up is not to critique these lab studies. We don't understand evolution in a fluctuating environment. We're still sort of at a stage where we are understanding evolution in sort of these artificial conditions created in lab. But in an ecological setting, the selection pressure is multidimensional because resources change, pathogens are present, antimicrobial agents are present, and time varying and intensity varying factors are also there. So what might we do in an ecological setting to understand evolution? We can't control the environment, but what we have to do is sort of try and understand what is happening in this ecological setting and try and infer by what is happening as to what does it tell us about the underlying evolutionary process which shaped it the way it did. And I'll just take a couple of minutes to mention two examples. First one is this paper that came out 10 years back from Josh Plotkin's group in Pennsylvania. It's about influenza A. Influenza is interesting in the sense that it comes every year. Every year infects a large fraction of people, and about a week or so of infection, it goes away. So our immune system takes care of that. But next year it infects again. Then the reason it infects next year is because it has come in a mutated form. And the selection pressure, the protein that our immune system recognizes is this hemagglutinin protein, which is called HA protein. And this is the structure of this protein, just representational purposes. So every year, this HA protein comes out mutated by having changed its amino acid sequence. What is interesting is that for starting 1980s, we have a lot of data available for the HA protein for influenza A virus. And this is just a phylogeny drawn of the variants of influenza that were present. I believe this is from late 80s to mid 90s. This is about a 10-year period. And in this 10-year period, there are a couple of things that stand out. One is that a lot of evolution has taken place in this 10 years. In fact, 20% of all amino acids in this protein have mutated in this decade, which means that the pressure on the protein is to change and evolve into something that is not immediately recognized by our immune system is actually huge. What Plotkin, so all this data is available, and this data is available for a large number of pathogens. So what Plotkin and co-workers do in this really nice study is that what they try and find out from this data is there any pattern to how changes are occurring here? Is it that when amino acid number 200 changes, amino acid number 500 changes very soon after that, and so on and so forth? If such patterns exist, they will tell us about they will help us predict the evolutionary trajectory of the protein as we go forward in time. And shown here is one such result, that what their analysis of all this data from a decade of sequences available tells us is that there is a site 344, this is amino acid number. Whenever 344 changes, 275 changes very soon after. What that means is that 344 changes, and now in a strain which is carrying this change at 344, if you introduce another change at 275, that strain becomes extremely virulent, easily spreads. And hence it's able to take over the population. And looking at this data, they're able to see many signatures of that particular interaction. So in this way, whilst we are still scanning the data to understand lessons from what has already happened in the past, the hope is to be able to enhance our understanding to an extent that we are also able to predict what might happen in the future. The second example of an ecological setting. Now remember, I mean, this virus example, for instance, we are not controlling the environment. The virus just is out there in the ecology. It undergoes all the selection pressure that the environment enforces it and comes back the next year. So we have no control over it. We are only trying to infer what has happened via these analyses. Second example is from the current pandemic, SARS-CoV-2. This was a paper that was published in, I think, August or September last year, where they solved the crystal structure of the S protein on the virus surface, which recognizes the ACE2 receptor in human cells. So this is RBD is the receptor binding domain of the S protein, which binds ACE2, and hence this permits the virus to gain entry into the mammalian cells. And very soon after this paper, David Baker's group published this paper in Science, which is an extremely elegant combination of theory and experiments. So what they did is that they recognized this surface. So they recognized that the surface where the receptor binding domain of the S protein on the virus is in contact with our ACE2 protein, that point of contact is very, very important. This is the key contact which establishes that, which enables the virus to gain access. And then through data analysis and through computations, what they design are just very short peptide sequence, artificially synthesized. They don't look like they don't look anything like ACE2. They design artificial peptides which theory predicts can bind to the receptor binding domain, and this binding of this amino acid chain, which is made in lab, prevents the receptor binding domain to interact with ACE2, because this binds so tightly that now this RBD has no space to it's been sequestered and it cannot bind the ACE2 receptor anymore. So when they test these receptors, when they test these peptides that their analysis predicts, by actually synthesizing these peptides, what they find is that they are able to prevent interaction between ACE2 and the ribosome binding domain at even very low nanomolar level concentrations of these peptides. This is a remarkable sort of exhibition of theory coming together and being able to really lead to some insightful results. This is the analysis that, but of course, what's going to happen here is that now, just as there was a huge pressure on the HA protein to come back having changed every year, there is now a huge pressure on the receptor binding domain to come back having changed and not be recognized by these peptides in this particular strategy, for instance. And if you see here, this is the mutational map of giving you an idea of all the mutations that are happening in the S protein since the last year and a half or so. And if you see, there are lots of changes taking place in the S protein sequence. The ones which are in orange are non-synonymous. So these are mutations which lead to a change in amino acid sequence of the protein, whereas the ones on blue are synonymous changes, which are mutations, changes in the DNA sequence, which do not lead to change in the amino acid sequence. And again, the hope with theory is that you might be able to sort of work with this data and try and recognize underlying patterns which dictate the evolution of this particular protein. And this is the work that, this is the analysis that an undergrad working with me has been doing in lab. Okay. So before I wrap up, I think I've gone 5 minutes overboard. What I want to summarize is that evolution by natural selection is an important mode of biological change. All the life diversity that we see on the planet is largely a result of action of natural selection. Evolution is a statistical process, and much of data analysis actually has its roots in evolution. Mendel's pea plant experiments that we've all heard of in the 1860s, they were largely not noticed by scientific community. In fact, in 1900, Mendel's paper was rediscovered, and it's called Rediscovery of Mendel. For the previous 34 years, people actually did not pay any attention to Mendel's work. And one of the reasons for that was that Mendel's work had a lot of data and math, and people didn't know how to reconcile this with biological understanding of what was going on. Francis Galton was a cousin of Darwin, and he was obsessed with making measurements and collecting data and trying to explain evolutionary phenomena. And again, 1870s and 1880s, because of his obsession with data measurements and coming up with ideas regarding how evolution proceeds, it led to a large number of very interesting debates in the second half of 19th century, post publication of Origin of Species. And Sewell Wright's work we just talked about, who proposed the metaphor of mountain climbing, these peaks and valleys shaping evolutionary processes. And what I hope to have communicated is that to understand evolution, we can do two approaches: do experiments in lab ourselves, or we can observe what's happening in ecology in terms of species spread, the diversity that we see, the mutations that happen, and try and make sense of it through theory computation or analysis of just enormous amounts of data, because sequencing now is relatively cheap. And if you're interested in sort of as a lay person or a student of the subject who's just starting out, I would strongly recommend that you go through these books or book. Many of these authors have written more than one book. So you might want to go through these names. Charles Darwin, Origin of Species is very easy to read. Vital Question, Nick Lane has written a bunch of fantastic books. If you're interested in the question of heredity and how it has been thought about since the last few hundred years, Carl Zimmer has this. It's a big book, but it's a fascinating read about how heredity has been thought, and it comes up all the way to CRISPR. Richard Dawkins has written a bunch of books, and Peter Bowler and David Common have again written more than one book. Just before I end, I chose not to talk about my work because I wanted to give you a larger perspective, but some of the questions that we are interested in my lab is understanding, and we do both experiments and theory. We don't study what is happening in ecology. We do experiments and theory to understand questions regarding, can we understand the process of speciation, where we have one species and it splits into two different species? That's called a speciation event. And we try and create conditions in labs such that speciation events will have, and we try and understand the underlying theory behind it. At the same time, we try and understand that how do species resist extinction by evolution of cooperative relationships, and that ties in with this theme of evolution of metabolic cooperation. Again, theory and experiments with this. And one of the theoretical questions that we're interested in is the evolution and maintenance of sexual reproduction, which is a separate topic. So, thank you for having me here, and I will be happy to take any questions. Should there be.
Thank you, Supreet. That was a really very interesting talk, looking at evolution to like from an angle of computational modeling and others was very interesting. I'm sure the picture that you showed in the first or second slide, maybe that Darwin had made in his notes, I think the new Darwins will write I compute. So let's, what I'll do, I'll not take much time. I'll just jump into the questions. We have some questions about the experiments that you showed in the beginning. Right. Anjana Nayorim asks, does the time taken for development of mutations is related to the dose of antibiotic that is used? Like if you change the range of the antibiotic with the number of mutations, or the time taken to get those mutations change?
Yeah. Well, I think that design is not trivial, because see, for a mutation to happen, a cell has to be able to divide. If it can't divide, mutation cannot happen. So if you give an antibiotic concentration that's very high, then it won't divide, and unless it divides, it won't pick up the mutation that makes it resistant. So typically how these experiments are done is that you give it a concentration of antibiotic which is below the MIC, and hence it picks up, so it won't kill the bacteria. MIC, for those of you who don't know, is the minimum inhibitory concentration which stops growth, is a minimum concentration that stops growth. So you give a concentration which is below MIC, and it picks up a mutation, and then for this mutated strain, MIC is higher, and then you jack up the concentration again, but again below the new MIC, and you keep doing this over and over again. The problem is that if you start off with a concentration that's too high, you're probably going to kill off the population, and it won't happen unless it's like very, very fortuitous event takes place. But when you add those of you who do experiments, if you add the wrong antibiotic to a tube, you never see growth, right? It's because the antibiotics that we work with, it's really high concentration.
So the another question that Anjana asked is, what is the hypothesis being tested in the Illinois corn oil experiment? And what will be the endpoint of this experiment, as this has been conducted for more than 100 years now?
Yeah, so yeah. That's a good question. Yes. See, historically, I think it's a very, very important experiment. It's an extremely important experiment, because what the linear increase shows is the fact. See, the linear increase is only possible if there are large number of loci which control oil production. If you have only one locus in the genome which controls oil production, then you will see step increases. That one locus spreads, then you are waiting for a new mutation to happen at this particular locus, and then oil production increases in a step function. Then you wait again for a new mutation to happen, and so on and so forth. The fact that it's linear shows us that the control of oil production in a corn plant is spread throughout the genome and is controlled by hundreds, if not thousands of loci which are spread throughout the genome. And this brings us now to sort of the quantitative trait loci, QTL type of work, or JIVAS type of studies that are being increasingly done, which look at identifying loci which control what are called complex traits, such as human height, for instance. So that experiment has been the first one to demonstrate that this is what's going on. So historically, it's a very, very important experiment. I think it's become a legacy experiment now. I don't know what is the next question that they can ask. So if you remember the graph that I showed you, in the middle of the experiment, they started selecting for low 15% also. So oil concentration went down, and then they started from there up. So they're playing with all of it. But I think it's a very important experiment in the history of the subject. It perhaps may not have a lot to tell us going forward in terms of new insights, but it's a legacy experiment.
So another student is actually again curious about the same experiment, and he asked, "Sir, could we get the data of this long-term experiment mentioned by you available? Is this available for the general public to experiment and play around with computational techniques?"
Yeah.
And
yeah. No, go ahead.
And he also asked that, does your lab provide such an opportunity for online experimentation?
To answer the first question, that so I talked about two long-term experiments, one a 125-year-old experiment and the other one 75,000 generation experiment. Most of these studies will have genome sequences which are publicly available. So I think if you go and look up the supplementary or the method section of these publications, you should be able to find those sequences. And nowadays, the data is available to the extent that the E. coli experiment, you will have 1,000 genome sequenced in a single paper. So a lot of that data will be available, and I think people are welcome to play with it. These are publicly available databases. Anybody interested in exploring evolution, or more importantly than that, learning more about evolution and how you can use computation to understand some of the things that I've talked about, you're very welcome to write to me, and I'll individually reply to you and see what we can do together. But yeah, I mean, half my group does theory and half my group does experiments.
So one of the question is about human evolution. Anant asks us that how many generations will it take for any mammal to branch out into a completely different species? If geographical isolation or any other type of reproductive isolation continues, after how many generations can we see speciation amongst humans?
I have no idea. We don't know. But what we do know is first of all, I think this definition species itself is very contentious. There is a lot of disagreement over what is a species. But the most commonly held is a definition that was proposed by an ornithologist called Ernst Mayr in the middle of 20th century. And Mayr says that two species are different species if the male from this and female from this cannot mate to produce a fertile offspring. So a horse and a donkey can mate, but a mule is sterile. Hence, horse and donkey are different species. Mm-hmm. So now we don't know. So the question is that if you have one population of one species, how long does it take for them to become reproductively isolated from each other, that they can't mate anymore? We don't know complex species because timescales become longer. But we do know that signs of reproductive isolation appear. This is experiments that have been done in 1960s, 70s, 80s. A lot of experiments in this regard were done with Drosophila. And we do know that signs of reproductive isolation appear as fast as tens of generations. More recently, there have been experiments with yeast which have shown that reproductive isolation can evolve in a few hundred generations. That is one of the questions that our lab works on also. But it's the timescale of if it's a timescale of a few hundred generations, unfortunately, the generation time is a relevant factor, and we don't know, because we can only work with species and answer these type of questions with species which have a very short generation time.
So one last question I'll take because there's not much time now. So Anurup asks us that it is seen that functions in a particular environment generally drives evolution. But if we see some organisms having extraordinary tolerance, which is not exactly required on Earth, like D radiodurans being able to tolerate extreme radiation, tardigrades having extreme tolerance to multiple stresses, and so on. What factors might have induced the development of these extraordinary capabilities?
Yeah, I mean, there could be a couple of explanations. I mean, one of the things is that you can never prove that this happened because of this particular reasons, because we can't go back millions or billions of years and recreate things. But this could happen because of two different reasons. One reason is what is called a selectionist reason, in which what we are saying is that maybe there was a selection pressure on the organism to develop that feature at the time when it was evolved. And later on with time, so somebody might become resistant to some form of radiation because Earth was getting a lot of radiation at the time. But later on, the radiation sort of disappeared, but the organism never really lost its ability for its tolerance towards radiation. So this is an adaptive explanation for what we see today in an organism. The other, which is a more interesting explanation, is that not everything happens for an adaptive reason. Some things happen just because of chance events. I'm reminded of this joke where they say that why do human beings have nose? They have a nose so that you can put spectacles on it. But that's not the reason why nose evolved, right? So evolution is not just linear. It's not goal oriented or anything. There is a lot of noise in this process also, and in that noisy process, a lot of stuff can happen. Also, I might go with the intention of buying a chocolate for myself, but along the way, if I like a bag of chips, I might buy chips also. So I didn't intend to buy chips. It's just that I was there, I bought it. So a lot of chance events happened, and some of these features could be an outcome of that as well. In 1979, there is a classical paper by Gould and Levontin, which is called Spandrels of San Marco, which talks about exactly that issue, that not everything that we see in an organism is because of natural selection. Chance events dictate evolutionary outcomes too.
Mm-hmm. So one last question that we asked to all our speakers, because we have, as you know, the audience is full of students and they are, pursuing different fields of science, of career, and many of them want to be data scientists, want to take up a field in data science. So how was your journey coming to, this field exactly, like evolution? You're a trained chemical engineer, and biology. I saw you have, lectures for biology for engineers. I mean, how did you, was this a chance event, or, how did you land up here? Or
it was totally a chance. No, it was totally a chance event. And when I went to do my PhD, I only went with one goal, that I will not do biology because it's boring. But then when I went through what the problems were being talked about, I realized that the only problems that sounded really cool to me were the problems of biology. And then I thought that because these problems are so interesting, I'm going to learn biology and whatever it takes, because I want to work on interesting problems. Now, if that means that I have to sit through a metabolism class, I'll sit through a metabolism class. If it means I have to sit through a computational biology bioinformatics class, I will sit through that also. So I feel that, I think, again, young people, you look for something that excites you. And once you find an exciting problem, then you learn and do whatever it takes to work on that problem. If it's experimental biology, you learn experimental biology. If it's hardcore data analysis, you learn hardcore data analysis. But what is that problem that is a really exciting problem that, excites you? And I find biology fascinating. So we keep learning,
Thank you. That's a great piece of advice for all our budding data scientists here. And thank you, everyone. I hope you have got the feedback form. And please fill the form and you'll get the certificate of participation, whoever wants to fill the form. And thank you, Dr. Supreet Saini. It was a great talk. And all of us enjoyed. I'm sure you're going to be bombarded with many emails from students from all over the country. And,
I look forward to receiving them, and I will reply to everyone. No problem whatsoever.
Thank you. Thank you, everyone. Thank you so much. And please stay tuned on our social media platforms. We keep announcing our upcoming programs, events, and the data science webinars that are going to come. Please do that. Thank you so much. Thank you, everyone.
Bye, everyone.